So if you look at patients dealing with advanced cancer, specifically cancer that's spread to the bone,
Yeah.
there's this one complication that's, well, it's almost universal.
Anemia.
Severe debilitating anemia.
Right.
And these patients are suffering from this just profound fatigue because their body, it simply can't produce enough healthy red blood cells.
And it's been a frustrating mystery, really, for decades in oncology. I mean, the bone marrow is ground zero for blood production. That process is called hematopoiesis.
Right.
But it's also, the perfect fertile ground for cancer cells to metastasize and grow. We've always just assumed the cancer was sort of crowding out the healthy cells.
Just a space issue.
A space issue. But the actual mechanical link between a tumor sitting in the bone and the systemic blood failure, that's been frustratingly murky.
So is it just a crowding problem, or is there something more specific, like a coordinated attack happening at the cellular level?
That's the question.
And that's what we are deep diving into today. Our sources reveal something pretty startling. The cancer isn't just crowding, it's pulling off a perfect targeted theft. And this theft promotes the tumor's own survival, while at the same time, it destroys the patient's ability to oxygenate their body.
Yeah.
So our mission today is to follow the iron, the most critical element for healthy blood, and understand how a specialized immune cell gets hijacked.
Hijacked by the cancer to cause both metastasis and anemia.
Okay, let's unpack this. The tumor's ingenious mechanism for survival.
So to really get how the cancer steals this iron, we first have to look at the normal infrastructure, the factory, so to speak.
The red blood cell factory.
Exactly. And it relies on these very specific specialized cells called macrophages.
Okay, but macrophages are usually the garbage collectors, right?
Yeah.
They eat debris, old cells. These are different.
These are very different. They are absolutely essential components of what the sources call erythroblast islands, or EB.
Erythroblast.
Yeah, you can think of it like this. Red blood cells are born in these little clusters, these islands, deep inside the bone marrow.
And at the center of each island
is one of these specialized macrophages. You could almost call it an iron nurse. It's surrounded by a cluster of developing red blood cells, which are called erythroblasts.
The nurse in the nursery.
A perfect analogy, because making red blood cells erythropoiesis is incredibly iron-intensive. Iron is what makes hemoglobin work.
So what's the iron nurse's main job in this island?
Its main job is recycling. When old red blood cells die, the macrophage engulfs them. It strips out the iron, and then it immediately turns around and transfers that recycled iron directly to the new erythroblasts that are clustered around it.
So it's the recycling center and the supply line, all in one.
Exactly. Without that nurse cell, the whole production line just collapses instantly.
That makes a lot of sense. So how did the researchers actually pinpoint this specific macrophage among all the other cells in the bone marrow?
Well, they used a combination of specific markers on the cell surface, things like VCM1, CD163, CCR3. It's a bit of a mouthful.
Right.
But what really matters is the powerful genetic signature they carry, a heme metabolism signature. It proves they are dedicated iron handlers.
Okay, so let's dig into that mechanism. What are the molecular tools that this nurse cell uses?
It has specialized molecular doors. To bring iron in, it expresses a gene called TFRC, which makes the transferrin receptor, or CD71.
Okay, so that pulls iron into the nurse cell.
Right. But crucially, for pushing that recycled iron out to the developing blood cells, it uses a gene called SLC40A1 that makes a protein called ferroportin, or FPN.
Ferroportin.
And FPN is the sole known iron exporter in vertebrates. It's the only way out. It's the dock that the nurse cell uses to push iron out to the erythroblast.
So FPN is the outward-facing iron dock, perfectly placed to feed the baby red blood cells.
Right.
And this macrophage population, which we can call MAC, had the highest EBI score in all the data, confirming its role.
Precisely. They are the specialists. But here's the first sign of trouble the sources revealed. When bone metastasis happens, these specialized MAC iron nurses, they're not where they should be.
They're not in the nursery.
They're gone. They are highly, highly enriched right in the bone metastatic niche, meaning they've physically relocated to be right next to the tumor cells, far away from the healthy erythroblasts they're supposed to be feeding.
So that spatial shift is the first major red flag. The cancer is basically putting out a recruitment call for the most iron-rich cells in the entire bone marrow.
And pulling the nurse away from the nursery.
So the iron nurses, packed with the recycled iron, are now clustered right next to the tumor. How does the cancer actually get them to hand over the goods?
Well, the tumor doesn't really ask. It exploits the macrophage's existing function by putting on a perfect disguise. So remember FPN, ferroportin, the iron exporter on the macrophage?
It's Doc pushing the iron out.
Well, now the tumor cell itself, and they confirm this with imaging and molecular analysis, starts to highly express CD71.
Wait, CD71, that's the iron importer. Isn't that the exact same receptor the normal developing erythroblasts use to pull iron in?
That is the crucial insight, exactly. The tumor cell acts like a giant hungry sponge. It puts on the same bib as the baby red blood cells.
Wow.
It positions itself right next to that FPN exporter on the macrophage and just overexpresses the CD71 importer, essentially hijacking the entire iron pipeline. The iron gets rerouted right into the tumor's metabolism, completely bypassing the starving erythroblasts.
But why does this type of tumor cell need so much iron? I mean, what makes it so critical for survival, specifically in the bone?
It's an adaptation to that specific environment. The bone marrow is inherently hypoxic.
Low oxygen.
Very low oxygen. Most cancer cells struggle there, but with this new huge supply of iron, the tumor cells can ramp up heme synthesis, which is critical for making energy, through oxidative phosphorylation.
So stealing the iron lets the tumor thrive in these suffocating low oxygen conditions.
It's a key survival tool. But the sources show the adaptation goes even deeper than just energy. The tumor starts to mimic the very cells it's starving. This is what they call erythroblast mimicry.
Erythroblast mimicry, what does that mean?
This is where it gets really fascinating. Because the tumor cells are now flush with iron and heme, and they're coping with this low oxygen, they start activating genes that are normally only found in red blood cell precursors.
Like what?
The most striking example is that these tumor cells start expressing hemoglobin beta, HBB, a core component of hemoglobin, the molecule that carries oxygen.
A tumor cell is making hemoglobin. That seems completely counterintuitive. What's the molecular switch for that kind of change?
It's the hypoxia itself. The low oxygen environment actually triggers the expression of an erythroid transcription factor called GATA1 inside the tumor cells.
And GATA1 is the master switch?
It's the master switch that tells a healthy cell, okay, time to become a red blood cell. So once GATA1 gets flipped on, it drives the production of all these globin genes, like HBB. By mimicking the erythroblast, the cancer cell gets this massive adaptive edge.
So if all the iron is being funneled to this hungry mimicking tumor, the original customer, the erythroblast, is left with nothing.
It's starved. And that perfectly explains the systemic consequence: cancer-associated anemia.
The local theft causes a total system failure. What did the researchers actually see in the blood of these metastatic mice?
Well, they saw all the classic signs of anemia: decreased hemoglobin, obviously, reduced red blood cell counts, and really critically, an increased red cell distribution width, or RDW.
High RDW. That means the red blood cells being made are all different sizes, right?
Right.
Sign of bad production.
Defective production, exactly. And when they looked closely at the erythroblasts still trying to grow near the tumor, they were basically screaming for iron. They showed this massive compensatory increase in their own CD71 expression, the iron importer, because they were just chronically iron deficient. But even with the door wide open, there was nothing coming in.
So they couldn't finish the job.
They couldn't. They had a reduced globin gene expression because they just lacked the raw material, the iron, to build the hemoglobin molecule.
It's like a profound quality control failure at the end of the assembly line.
That's a perfect way to put it. And the failure really shows up in the final stage, a process called enucleation.
That's where the cell kicks out its nucleus to become a mature red blood cell.
Right. That critical transition was severely impaired. The cells got stuck. They arrested in that final stage of maturation. The whole erythroblast island structure was just breaking down. The nurse cell was gone.
Now, this whole incredible model hinges on this one specialized macrophage being the linchpin. Did the researchers prove that if you get rid of that macrophage, the whole cascade stops?
They did. This was the smoking gun. They used these genetically modified mice called JDI mice.
JDI mice?
Yeah, it stands for just EGFP death-inducing. It allowed them to very specifically find and eliminate just those MAC iron recycling macrophages and nothing else.
Okay, so if the theory is correct, taking out the macrophage should hurt the tumor, help the patient's blood counts.
And that is exactly what happened. Depleting these specialized macrophages led to two things at once. First, impaired tumor growth, and second, a significant reversal of the decline in hemoglobin levels.
Wow.
It confirmed that this one cell is essential for both the metastasis and the anemia it causes.
Which immediately points to a therapeutic strategy. Did they also confirm it was specifically the iron transport?
They did. They used a drug called Vamiport, which inhibits FPN, the iron exporter on the macrophage.
So it blocks the duct.
It blocks the dock. The macrophage can't pump the iron out anymore, and its tumor-promoting activity just vanished. They also showed that simple iron chelators drugs, like DFO, that just mop up available iron, also slowed down tumor growth. The iron pathway is the absolute link.
Amazing. Okay, before we get to the human side of this, one quick question. How does the tumor get the macrophages to come over in the first place? What's the welcome mat?
The tumor cells are, they're sophisticated communicators. They're basically putting up a neon sign. They express these specific chemokines like CCL5 and growth factors, especially MCSF and IL-34.
Powerful recruitment signal.
Very powerful. And the MAC cells have the right receptors like CCR3 and CSF1R, so they just follow that chemical trail right into the metastatic niche.
So this whole elegant mechanism of theft and mimicry, it works in the lab. Is there solid evidence this same crime is happening in human patients?
Absolutely. When they analyzed human bone metastases from breast cancer, lung, kidney, they found macrophages with that exact same high iron metabolism signature, high FPN, high HMOX1. The mechanism is conserved.
And the really weird part, the tumor cells making hemoglobin.
And that was also there. Tumor cells in the human samples were expressing both HBB and the iron importer, CD71. But here's the really critical piece of data. When they looked at patient data, high HBB expression in a primary breast tumor was associated with a significantly higher risk of developing bone metastasis. But, and this is key, not metastasis to the lung or the liver or the brain.
So that strongly suggests HPB expression isn't just a general marker of aggressiveness. It's a specific adaptation for surviving in the bone.
Precisely.
So what does this all mean for you, the listener, and for where treatments might go? I mean, if we connect this all together, it feels like we suddenly have these new dual-purpose targets.
That's exactly right. We have targets that could potentially treat the tumor and the debilitating anemia at the same time.
Yeah.
You could attack the adaptation itself, block the tumor's iron importer, CD71, or maybe inhibit the synthesis of hemoglobin inside the tumor.
Or you could go after the niche.
Or go after the niche. Try to prevent the initial recruitment of these macrophages. Or use a drug like Vamiport to block the iron transfer, which should, in theory, restore the iron nurse's function back to the healthy erythroblasts.
This really broadens our whole view of the bone metastatic niche, doesn't it?
Yeah.
We've spent so much time focused on the osteoclasts and osteoblasts, the bone-breaking cells.
Right, the bone destruction itself. But this shows how critical the immune system and metabolism are for the tumor survival there. It connects tumor biology to hematology in a way we've really needed for a long time.
The ultimate irony here, I think, is that the tumor has figured out how to hijack a system that evolved for life support, this essential recycling of iron for blood to fuel its own destructive survival.
Leading to a catastrophic failure of the host's own system. It's a remarkable and terrible piece of parasitic co-option.
So this study focused mostly on the cells directly handling the iron. But the data also showed that basophils, another type of immune cell that's known to secrete things like IL-4, were also highly enriched right there in the niche.
So here's a final thought for you to explore. If basophils are also in the niche, and we know they influence other immune cells, how might this basophil macrophage crosstalk? Maybe through a factor like IL-4, how might that further influence or even accelerate this whole iron theft process? Could blocking that kind of cytokine signaling be the next step in creating more powerful combinatorial immunotherapies for bone metastasis?